This lecture is going to cover chapter 3. It is going to be all about cells and tissues. So the first thing that we're going to talk about is a general overview of what cells are and what components make up a cell. So cells are going to be the structural unit of all living things. The human body is going to have about 50 to 100 trillion cells, which is a whole lot. All of those cells are going to work together to make everything function. So we base all of the information that we know about cells on something called cell theory. And what cell theory says is that a cell is the basic structural and functional unit of living organisms, that the activity of an organism is going to depend on the collective activities of its cell, and that the biochemical activities of cells are going to be dictated by their structure. You've probably heard the phrase before that form follows function. So the structure is going to determine what the cell will do. And then lastly, cell theory says that the continuity of life is going to have a cellular basis. So that means as we're talking about reproduction, replication, that kind of thing, it's all going to go back to the cells. So let's talk a little bit about the anatomy of the cell. So in general, a cell is going to have three main regions or parts. That's going to be the plasma membrane, the nucleus, and the cytoplasm. And then within all of those, there will be more constituent parts that we're going to talk about. Let's talk about the plasma membrane first. So the plasma membrane is going to be a transparent barrier that's going to enclose all of the cell contents. It is going to separate the things inside the cell from the surrounding outside environment. We refer to the plasma membrane as having a fluid mosaic model. That means it lives somewhere between a solid and liquid, so it's flowing, right? It's fluid, but it is definitely not necessarily a liquid. It's more of obviously a plasma type situation. So it's going to be composed of two layers of phospholipids arranged tail to tail. We will talk about why this is so important on the next slide. It's going to have cholesterol and proteins scattered throughout the phospholipid bilayer, and then oftentimes these cholesterols and proteins will have sugars attached to them. When that happens, we will refer to them as glycoproteins and glycolipids, because anytime we have a sugar, we get that prefix glyco. So the way that the phospholipid bilayer arranges itself is that if you think back to phospholipids, you'll remember that they have hydrophilic, so water-loving heads, and hydrophobic, or water-fearing tails. So in the phospholipid bilayer, there's going to be two layers of these. The hydrophilic heads are going to be oriented on the inner and outer surfaces of the membrane, and those hydrophobic tails are going to face each other, kind of like this, where the tails go to the inside. The purpose of this is to create an electrochemical gradient to where we are able to move some things across that plasma membrane, but it will allow the cell to kind of control what can go in and what can go out. So the role of proteins on the phospholipid bilayer, there's going to be several different specialized membrane functions. Proteins can act as enzymes, they can act as receptors for hormones or other chemical messengers, and they can also act in transporting. Whenever we need something transported across that electrochemical gradient, they can work to be channels or carriers to make that happen. Those sugars, so those glycoproteins that we talked about, are going to be branch sugars attached to the proteins. And they are going to abut the extracellular space. That means they're going to back right up to it so that they can interact with that extracellular space. The glycocalyx is going to be a fuzzy, sticky, sugar-rich area that is going to make up the cell surface. So that is going to also help a little bit with that electrochemical gradient and also help with that enclosing that we were talking about that the cell membrane does. The cell membrane is also going to have junctions. So cells are going to be bound together, and here we're talking about cells to cells, right? Junctions between adjacent cells. Cells are going to be bound together in three different ways. Glycoproteins in the glycocalyx can act as kind of like an adhesive, right? Because again, this is made of sugar. If you've ever gotten sugar wet, you know that it gets really sticky. So it can make these cells literally stick together like glue. There are also wavy contours of the membranes, and sometimes those adjacent cells will fit together like in a tongue and groove or a lock and key type fashion. And there can also be special cell membrane junctions that are formed. These will vary structurally depending on their roles. So again, form follows function. Here's a few of the different types of junctions that we can have. We can have tight junctions. These are going to be mostly impenetrable. These are going to bind cells together into leak-proof sheets. So things aren't really going to be going back and forth between them very often. The plasma membranes are going to fuse like a zipper to prevent those substances from passing back and forth through the extracellular space between cells. We also, so this is an example of a tight junction, we also have desmosomes, and we call these anchoring junctions. These are going to act like rivets that are going to prevent cells from being pulled apart as the result of mechanical stress. So it is literally going to be like a button and buttonhole type situation. We also have gap junctions. Gap junctions are so important because these are going to allow communication between our cells. These are typically hollow cylinders of proteins, and then the molecules can travel back and forth through these channels. So let's talk a little bit about the nucleus. The nucleus is going to be the control center of the cell. It is going to contain the genetic material, which is going to be our DNA. DNA is going to be needed to build proteins, and DNA is also necessary for cell reproduction. There is going to be three important regions. The nuclear envelope, which is going to be this area surrounding the outside of the nucleus, the nucleolus, which is this darker inner area, and then chromatin, which are these little threads that are going throughout, and that is going to be where our DNA is actually living. So let's talk a little bit about the nuclear envelope, the nucleolus, and chromatin. The nuclear envelope, again, is going to consist of a double membrane that kind of goes around and encloses the nucleus. It's going to contain nuclear pores. If you see all these little orange spots, these are nuclear pores that are going to allow for exchange of materials with the rest of the cell. So sometimes we need to move things in and out of the nucleus, especially genetic material. So the pores are going to be how we do that. And this encloses the nucleus in a jelly-like fluid called nucleoplasm. The nucleolus is going to be this darker area on the inside. It is going to, so the nucleus is going to contain either one or more of these dark staining nucleoli. These are going to be the sites of ribosome assembly. And the way that this happens is the ribosomes are going to migrate through these nuclear pores to make that protein synthesis happen. Another important thing to care about here, if you see these little darker lines running throughout, that's our chromatin. Chromatin is going to be composed of DNA wound around histones, and we will talk a little bit more about this later. It's going to be scattered throughout the nucleus, and we can see it when the cell is not dividing. So that is where our actual DNA lives. Let's talk a little bit about the cytoplasm. So the cytoplasm is going to be the cellular material outside of the nucleus and inside of the plasma membrane. So it's going to be all of everything in here. Okay. This is going to be the site of most of our cellular activities. This is going to include cytosol, inclusions, and organelles. There's going to be three major components of the cytoplasm, and it is going to be what I just said, the cytosol, inclusions, and the organelles. So what the cytosol is it is going to be a fluid that suspends other elements and contains nutrients and electrolytes. So your cell essentially is just going to have this substance inside of it that is a fluid-like substance where everything else lives, and that is your cytosol. We also can have inclusions. These are going to be chemical substances such as stored nutrients or cell products that are going to float around in that cytosol, and then we'll have organelles, right? These are things like our mitochondria, our endoplasmic reticulum. These are going to be the metabolic machinery of the cell that are going to perform those functions for the cell. Let's talk about mitochondria. This is one of our organelles. You've definitely heard that the mitochondria is the powerhouse of the cell. That is because this is what is making our ATP and making us have energy for all of the things that our body needs to do. The mitochondrial wall is going to consist of a double membrane with cristae, that's these little empty spaces on the inner membrane. The purpose of that is to increase surface area so that it can carry out the reactions in which oxygen is going to be used to break down food into ATP, right? So this is the guy that is actually taking our food and turning it into the energy that we need to make our body go. Ribosomes are also important. We will talk a whole lot about ribosomes. They have a lot of important jobs. So ribosomes are going to be made of protein and ribosomal RNA. These are going to be the sites of protein synthesis in the cell. These are going to be found in two different locations, and this is important. They can be free-floating in the cytoplasm, and they can also be attached to the rough endoplasmic reticulum. When we talk about, we will have a smooth and a rough endoplasmic reticulum. When we talk about the rough endoplasmic reticulum, it is rough because the outside of it is dotted with these ribosomes to almost make it look like sandpaper. So that is why it is called rough. So the endoplasmic reticulum is going to be fluid-filled tunnels, kind of like stacks and stacks and stacks of little canals that are going to carry substances within the cell. It is going to be continuous with the nuclear membrane, and what that means is that it is just going to hug all the way around the outside of that nucleus. And I put you a little table here, the difference between rough and smooth. So while your rough is going to be studded with ribosomes, your smooth is not going to have any ribosomes on it. It will lack ribosomes. Your rough functions to synthesize proteins, while your smooth is concerned with lipid metabolism. And then also your rough is really important in transporting, making transport vesicles to move proteins within the cell, while your smooth is really concerned with detoxification of toxins and drugs that get into your system. I put you a little table here that shows kind of how the synthesis and export of a protein happens by the rough ER. So essentially the first thing that is going to happen is as the protein is synthesized on the ribosome, it's going to migrate into the rough ER tunnel system. So here's your little ribosome. Here's the little protein that it's making, and it is migrating into the tunnel system. Then once it's in the tunnel, the protein is going to fold into its functional shape. Remember we talked about the primary and secondary and tertiary and quaternary structure of protein folding. This is what we're talking about right here. Sometimes we will attach short sugar chains to the protein, making it a glycoprotein at this point. Then it is going to move to stage 3. At stage 3, the protein is going to be packaged in a tiny little membrane sac that is called a transport vesicle. And then that transport vesicle is going to bud off from the rough ER and travel to the Golgi apparatus for further processing. So what is the Golgi apparatus? It is going to appear as a stack of flattened membranes associated with tiny vesicles. The purpose of the Golgi apparatus is to modify and package proteins arriving from the rough ER via those little transport vesicles. So it's going to produce a few different types of packages depending on where it's coming from. It's going to have secretory vesicles in pathway 1. It will have proteins and lipids in pathway 2, and it will have lysosomes in pathway 3. So depending on which way it goes depends on what you end up coming out with as your end product. So lysosomes, I mentioned that, but we don't know what that is yet. So lysosomes are just going to be membranous bags that are going to contain digestive enzymes. These enzymes can digest worn out or non-usable cell material to just kind of get that trash out of the cell so it's not taking up space or doing damage. These are also going to house our phagocytes, and our phagocytes come in and literally eat up debris. They engulf them and then they carry them away. So they're going to house those phagocytes that dispose of bacteria and cell debris. We also will have peroxisomes, and these are membranous sacs of oxidase enzymes. These are going to detoxify harmful substances such as alcohol and formaldehyde and anything toxic to us. They break down free radicals, which are highly reactive chemicals in our system. And after that process, the free radicals are converted to hydrogen peroxide, which is why these are called peroxisomes. And then after they become peroxide, they are then turned into water. They replicate by pinching in half and just kind of budding off, kind of like if you've ever seen something like sea sponges reproduce. It's really similar to that. The next thing we're going to talk about is the cytoskeleton. This is a network of protein structures that are going to extend through the cytoplasm. Its function is to provide the cell with like an internal framework that is going to determine the cell shape. It's going to help support the organelles, and it's going to provide the machinery for intracellular transport. So you want to think of the cytoskeleton kind of like the bones of the cell, essentially. There's going to be 3 different types of elements that form the cytoskeleton: microfilaments, intermediate filaments, and microtubules. And I put a little table there for you all to be able to see like illustrations of those, but also like microscopic images of them to kind of get a better idea of what they look like. The last thing as far as cell components we're going to talk about is going to be the centrioles. These are going to be rod-shaped bodies made of 9 triplets of microtubules. The purpose is going to be to generate microtubules, and they are responsible for the formation of the meiotic spindle during cell division. I also put a table in here just to help you all study that goes over all of the different organelles that we talked about during this part of the lecture. So that is what these slides are all about. Let's talk a little bit about cellular extensions. So cellular extensions are going to be surface extensions found in some cells. These are going to be things like cilia and flagella. Cilia are going to move materials across the cell surface. These are things like the little hairs that are located inside of our respiratory system to help move mucus out. Flagella are going to be things like the tail on the back of a sperm. The only flagellated cell in the human body is going to be sperm. And then we're also going to have microvilli, and microvilli are like tiny little finger-like extensions, and they come actually out of the plasma membrane, and the function of microvilli is to increase the surface area for absorption. So let's talk a little bit about cell diversity, because there's a lot, and this is not going to encompass all of the kinds of cells that there are in the human body. This is just going to cover a few that will definitely be important for this class. So the human body is going to have about over 200 different cell types. These cells are going to vary in size, shape, and function. We can have cells that connect body parts. These are going to be things like fibroblasts. Fibroblasts secrete cable-like fibers to be able to connect things together. We also have erythrocytes. Erythrocytes are also called red blood cells. The purpose of these is going to be to carry oxygen in the bloodstream. We're also going to have things like epithelial cells. Epithelial cells are going to cover and line bodily organs. So these pack together in sheets, and then they have these little intermediate fibers that are going to kind of resist tearing during, like, when these sheets are rubbing or pulling on each other. We're also going to have cells that can move organs and body parts. So these are going to be things like skeletal muscle and smooth muscle cells. And these are, at the most basic level, just like contractile filaments that are going to allow cells to shorten and lengthen forcefully to create that movement whenever we need it. We can also have cells that store nutrients. This is going to be something like adipose cells. So we can have these little lipid droplets that are stored in the cytoplasm so that we have that energy available whenever we want to use it, but we don't necessarily have to have it taking up space all the time, because we can condense it down and only use it whenever we actually need it. We're also going to have cells that work in our immune system to help fight disease, right? These are going to be white blood cells like our macrophages that are going to track down and digest those infectious microorganisms. And we can also have cells that are going to gather information and control body functions, right? These are going to be things like nerve cells or neurons. These are going to receive and transmit messages to other body structures. We're going to have cells of reproduction too, right? We have oocytes. That's just the very fancy science word for egg. This is going to be the largest cell in the body, and this is what divides to become an embryo upon fertilization. Then we can also have sperm cells. These are built for swimming to the egg for fertilization, and the flagellum in that case acts as the modal whip to move them along. And again, we just talked about flagellum and how the sperm is the only cell in the body that has flagella. So let's talk a little bit about cell physiology. So cells are going to have the ability to metabolize, digest food, dispose of waste, reproduce, grow, move, and respond to stimuli. We're going to talk a little bit about how they do this. We're going to start with membrane transport. So here's a little bit of important vocabulary that if you're not super familiar with these words, you may want to review them. You've definitely probably seen them before in an intro bio class. So a solution is going to be a homogeneous mixture of two or more components. A solvent is going to be a dissolving medium present in a larger quantity. So the body's main solvent is water. Most of the time, if you have a solvent, it's water, but not always. And then solutes are going to be the components in smaller quantities that we're kind of dissolving into that solution. We're going to have intracellular fluid, right? So within the cell, we're going to have nucleoplasm, we're going to have cytosol. These solutions are going to contain gases, nutrients, and salts all dissolved in water. We're also going to have extracellular fluid. We refer to this as interstitial fluid when we're talking about human anatomy. This is going to be fluid on the exterior of the cell, in between other cells. It's going to contain thousands of ingredients. The most common ones are going to be nutrients, hormones, neurotransmitters, salts, and a whole lot of waste products that the cell has pushed outside of the plasma membrane. We know that because of the hydrophobic and hydrophilic nature of the plasma membrane, that it is going to be a selectively permeable barrier. That means that some materials are going to pass through while others are going to be excluded. For example, nutrients can enter the cell, and we can keep undesirable substances out, and we can also use that electrochemical gradient to only pull in the ions that we want while we push out the ones that we don't. There's going to be two basic methods of transport that are going to be important for the sake of this class. We're going to have passive processes in which substance are going to be transported across the membrane without any input from the cell because they are working with that electrochemical gradient. And then we're going to have active processes. For the active processes, the cell is going to provide the metabolic energy in the form of ATP to drive that transport process. So our passive processes are going to be diffusion and filtration. In diffusion, molecule movement is going to be from high concentration to low concentration, and it's going to move down that concentration gradient. Particles are going to tend to distribute themselves evenly within a solution because the kinetic energy, which is our energy in motion, right? It is going to cause the molecules to move about randomly. And they all want to have their own little area where they have as much space as possible. And so that causes them to kind of repel each other until they reach the state of uniformity. The size of the molecule and the temperature are going to drastically impact the speed of diffusion. So just know that. Molecules will move by diffusion if any of the following applies to the situation. So if the molecules are small enough to pass through the membrane's pores, like those channels that are formed by membrane proteins, or like our nuclear pores that we talked about earlier, if the molecules are lipid soluble, then they're not going to have any problem at all moving by diffusion. And if the molecules are assisted by a membrane carrier. So we're going to have a couple of different types of diffusion. We will have simple diffusion. This is going to just be the unassisted movement of solutes. Solutes are going to be lipid soluble or small enough to pass through the membrane's pores. And we can also have osmosis. So osmosis is going to be the simple diffusion of water across a selectively permeable membrane. So because water is highly polar, those molecules are going to easily cross the plasma membrane through aquaporins. And that water is going to move down its concentration gradient. So let's talk a little bit about how osmosis applies within the body. So we have what we call isotonic solutions. Whenever you go to the hospital and you get IV fluids, those are isotonic solutions. These are going to have the same solute and water concentration as the cells. So the cells are going to gain and lose water at the same rate, so they will stay in a state of homeostasis. You can also have hypertonic solutions. These are going to contain more solutes than the cells do. So what this causes is for the water to be drawn out of the cell. You can also have hypotonic solutions. These solutions are going to contain fewer solutes, so they're going to have more water than the cells do. In this situation, the water is going to want to be pulled into the cell, so it will make the cell expand until sometimes it explodes. You can also have facilitated diffusion. In facilitated diffusion, you're going to have something transporting those substances. So this usually transports lipid insoluble substances, charged substances, and substances that are way too large for passage through the membrane pores. Examples of substances that move by facilitated diffusion are going to be things like glucose and chloride ions. So oftentimes those protein membrane channels that we talked about earlier, or protein molecules are going to act as the carriers in these situations. You can also have passive processes like filtration. In this situation, water and solutes are going to be forced through a membrane by essentially hydrostatic pressure. So the pressure gradient has to exist to push the solute containing fluid from a high pressure area to a lower pressure area. Filtration is critical for the kidneys to work properly. We will spend a whole lot of time talking about filtration whenever we do the urinary system. Let's talk a little bit about active processes. In active processes, ATP is going to be used to move substances across a membrane. Active processes are going to be used when substances are way too large to travel through membrane channels. If the membrane lacks special protein carriers for the transport of certain substances, if substances are not lipid soluble, or if substances have to move across a concentration. We're going to have active processes as well. The two most important types are active transport and vesicular transport. So in active transport, amino acids, some sugars, and ions are going to be transported by protein carriers known as solute pumps. ATP is going to energize these solute pumps to make it to where they can do their jobs. And in most cases, substances are going to be moved against the concentration or electrochemical gradients. A really good example of this is going to be something like a sodium-potassium pump. This is necessary for nerve impulses. It's really important. It also has a large role in the cardiac system as well. So in this situation, sodium and potassium are going to be pumped against their concentration gradients. Sodium is going to be transported out of the cell, and potassium is going to be transported into the cell. Vesicular transport is going to be another active process, and in this process, substances are going to be moved across the membrane in bulk without actually crossing the plasma membrane directly. So this is going to be a situation where they're going to be enveloped in a vesicle, and then that vesicle is going to move them across. So this is going to be things like exocytosis, endocytosis, and within endocytosis is where we get phagocytosis and pinocytosis. Phagocytosis is whenever something like a macrophage is engulfing something to then move it out of a cell. Pinocytosis is exactly the same thing, except instead of moving a solid thing, you're moving liquid across. Sorry, I got too excited and I told you about these slides already. So exocytosis, is going to be the mechanism that cells use to actively secrete hormones, mucus, and other products. In this situation, like I said before, material is going to be carried in a membranous sac called a vesicle that is going to migrate to and combine with the plasma membrane. Contents of the vesicles are going to be emptied to the outside from there. And I included a little diagram here for exocytosis. We're looking at pathway 1 right here. In endocytosis, extracellular substances are going to be enclosed or engulfed in a membranous vesicle. The vesicle is going to detach from the plasma membrane and move into the cell. Once in the cell, the vesicle is typically going to fuse with a lysosome, and then the contents are going to be digested by those lysosomal enzymes. In some cases, the vesicle is going to be released by exocytosis on the opposite side of the cell. So phagocytosis literally means cell eating. So a cell is going to engulf a large particle such as a bacteria or just cell trash, dead body cells, things like that. And it is going to move it out of the cell. And these are all protective mechanisms. So phagocytosis is a protective mechanism to move things out, not a means of getting nutrients. Penocytosis literally means cell drinking. It's the same thing as phagocytosis, except the cell is going to essentially like drink droplets of extracellular fluid containing dissolves proteins or fats or whatever it is that we want to get out of there. The plasma membrane is going to form a pit, and then the edges are going to fuse around the droplet or fluid. It is a routine activity for most cells, especially those involved in absorption. So like this is happening all the time in the small intestine. We can also have receptor-mediated endocytosis, and this is going to be a method for taking up specific target molecules. Receptor proteins on the membrane surface are going to bind only to certain substances. The highly selective process of taking in substances such as enzymes, hormones, cholesterol, and iron, again, will only really occur on that surface membrane. Let's talk a little bit about cell division. For this one, I actually put a couple of videos in here for you guys because this is one that you're going to want to go back and do over and over and over again. Because for cell division, it takes a few runs at it to really get it. You're not going to be able to go through one lecture or watch one video and get it, and you can study on paper, and that is really, really helpful. But sometimes it helps to just watch those processes happen over and over and over again. So I encourage you to watch those, and then when you come to lab on Monday, or if you want to meet before, we can sit down and really get into the nitty-gritty if it's just not sticking for you. So let's talk a little bit about protein synthesis. So DNA, we know, is going to serve as a blueprint for making proteins. We know that a gene is going to be a DNA segment that is going to carry that blueprint for building one protein or one polypeptide chain. Proteins are going to have many functions, right? We talked about this in the last lecture. We talked about how we have fibrous or structural proteins that are going to be for the sake of building materials for cells. And we also have globular or functional proteins that can act as enzymes or are a biological catalyst. So we know that DNA information is going to be coded into sequences of bases. A sequence of 3 bases is going to code for an amino acid. So most ribosomes, we know that the ribosome is the manufacturing site of proteins. Most of them are going to be located in the cytoplasm, except for those that live on the rough ER. DNA actually never leaves the nucleus in interphase cells. DNA is going to require a decoder and a messenger to carry instructions to build proteins to the ribosomes. So both the decoder and the messenger functions are carried out by RNA. There's going to be 3 different varieties of RNA that I want you to pay attention to for the sake of this class. Ribosomal RNA, or rRNA, is going to help form the ribosomes where the proteins are built. Messenger RNA, or mRNA, is going to carry the instructions for building a protein from the nucleus to the ribosome, and transfer RNA, or tRNA, is going to escort appropriate amino acids to the ribosome to help build that protein out. So protein synthesis is going to involve two major phases, transcription and translation. And I put you a little diagram to help explain this better. In transcription, transfer of information from DNA's base sequence to the complementary base sequence of mRNA is going to happen first. The DNA is the template for the transcription, whereas mRNA is the product. Each DNA triplet is going to correspond to an mRNA codon. And if the, so like for instance, if the DNA sequence is AATC, GTTCG, then the mRNA corresponding codons are going to be UUA, GC, AA, GC, right? So we know that these codons correspond, and we know that 3 of them are going to correspond to a very specific amino acid. So when you start building up 3 and 3 and 3 and 3, then you end up with a recipe for a protein. In translation, the base sequence of the nucleic acid is going to be translated to an amino acid sequence. So amino acids, we know, are going to be the building blocks of protein, right? So the purpose of translation is to make those building blocks. This is going to occur in the cytoplasm and is going to involve 3 major varieties of RNA. In the first step, mRNA, so our messenger RNA, is going to leave the nucleus through those nuclear pores and attach to the ribosome, and at that point translation is going to begin. In step 2, the incoming transfer RNA is going to recognize a complementary messenger RNA codon calling for its amino acid by temporarily binding its anticodon to the codon. At step 3, the ribosome is going to move along the mRNA, and a new amino acid is going to be added to the growing protein chain. After that, the released tRNA is going to re-enter the cytoplasmic pool and be recharged with a new amino acid. So let's talk a little bit about tissues, because tissues are going to be very important for this class. I know we talked about them previously, but we're going to dive a little bit deeper in. So tissues are going to be groups of cells with similar structure and function. We're going to have 4 primary types: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Let's talk about epithelial tissue first. So epithelial tissue, the locations where we're going to find it. We're going to find it on body coverings, body linings, and in glandular tissue. It functions for protection, absorption, filtration, and secretion. The hallmarks of epithelial tissues are that they cover and line body surfaces. They're often going to form sheets with one free surface. We call this the apical surface, and an anchor surface, which we refer to as the basement membrane. They are avascular, which means they have no blood supply. We have blood that will diffuse up into that tissue, but it's not because there's vessels within it. It is because the vessels stop and then it's just kind of flowing out. It can regenerate really, really easily if it's well nourished and moisturized and taken care of. So I put you a few different tables to look at here and a few different examples of these cell types. The way that we're going to classify epithelial tissue is going to deal with the number of cell layers and the shape of the cells. So when we're talking about simple cells, we're talking about one layer. So if we say like simple squamous, we're talking about one layer of squamous cells. If we talk about the epithelium being stratified, that means there's more than one layer. So those both deal with number of cells. When we're talking about shape of cells, we can have squamous shells, which are going to be flattened. These are going to look a little bit like fish scales under the microscope. We can also have cuboidal cells. These are going to be cube-shaped. These are going to look like little dice under the microscope, and we looked at a lot of these on Monday. And then we can also have columnar cells. These look like little columns. So for simple squamous epithelium, The location is going to be usually within membranes. This is what's going to form our membranes. It's going to line the air sacs of the lungs, it's going to form the walls of the capillaries, and it's going to form our serous membranes that line and cover organs in the ventral cavity. The purpose of simple squamous epithelium is for filtration and diffusion. Simple cuboidal epithelium is going to be a single layer of cube-like cells. We will find this oftentimes in glands and in their ducts. This is going to form the walls of the kidney tubules and also cover the surface of the ovaries. For simple columnar epithelium, you will notice that this is going to be a single layer of tall cells, and you'll see these big divots. Let me point out some of these to you. You'll see these big divots in the surface. We call these goblet cells. The purpose of goblet cells is to secrete mucus. So we will find these places where mucus is found. So this is going to be places like the lining of the digestive tract and mucous membranes, right? So any body cavity that is opening to the exterior naturally, we will find these cells there. We can also have pseudostratified columnar epithelium. And on this, all the cells are going to rest on a basement membrane. It is going to be a single layer, but because some cells are going to be shorter and some are going to be taller, and you may even have a difference in nuclei placement, it's going to look stratified until you take a closer look and see the outlines and see that you only have the one layer of cells there. And we looked at these in lab as well. We're going to find these in the respiratory tract because they're often ciliated. So ciliated cells are those hair-like cells that are going to help us cough and move things up out of our respiratory tract. And these function in absorption and secretion. So our stratified epithelia is going to consist of 2 or more cell layers again, and is going to function primarily in protection. So we will see stratified squamous epithelium. These are going to be named for the cells present at the free surface, which are squamous, and the functions of stratified squamous epithelium is protective. So this is going to be a protective covering where friction is really, really common. This is going to be the kind of cells that you find on our skin. So because our skin is getting constant friction from the outside environment. You'll also find this in the mouth and the esophagus because we lose a lot of cells in that area due to eating. Stratified cuboidal epithelium is just going to be 2 layers of cuboidal cells. And stratified columnar epithelium, in that the surface cells are going to be columnar and the cells underneath can sometimes vary in size and shape. The stratified cuboidal and columnar are going to be really rare in the human body. We're only mainly going to find them in ducts of very large glands. We also have transitional epithelium. This is a really special type of epithelium. This is going to be composed of modified stratified squamous epithelium. The shape of the cells is going to depend on the amount of stretching, but they often look like little umbrellas under the microscope. And it functions in stretching and has the ability to return back to normal shape. So this is going to be for organs that expand and then shrink down. So like our urinary bladder is composed of transitional epithelium. We can also have glandular epithelia. 1 or more cells responsible for secreting a particular product together is going to be classified as glandular epithelia. The secretions are often going to contain protein molecules in an aqueous fluid. Aqueous just means water-based. And the secretion is going to be an active process. So these are really important and cause 2 of our major gland types to develop from these epithelial sheets. This is where we get our endocrine glands. These are going to be ductless. And their secretions, which are hormones, are going to diffuse into blood vessels. So examples of this are going to include like our thyroid, adrenal, and pituitary glands. We can also have exocrine glands. These secretions are going to empty through ducts, and these are going to include like our sweat and oil glands, the liver, the pancreas, things like that. Let's talk a little bit about connective tissues now. So connective tissues are going to be found everywhere in the body, and the purpose is to connect body parts. This is going to include the most abundant and widely distributed tissues. They're going to function in protecting, supporting, cushioning, and insulating. Types of connective tissues are going to be things like bone, cartilage, dense connective tissue, loose connective tissue, and even blood. So we've all heard of bones before. This is also referred to as osseous tissue. This is going to be composed of osteocytes or bone cells, sitting in lacunae cavities. Lacunae just means a little cave. So there's these little holes that you can see around, and their purpose is to hold these little osteocytes. The bone is going to be made up of a hard matrix of calcium, and it's going to have a large number of collagen fibers. The purpose of bone is to function to protect and support the body, and we will obviously be talking way more about this whenever we get into the skeletal system, but I'm just giving you the overview for now. We also have cartilage. Cartilage is going to be less hard and more flexible than bone. This is going to be what we find like in our nose and our ears, that's a little bit bendy but not quite bone. This is going to be mainly composed of chondrocytes. This is the cell that makes cartilage. And we're going to have a few different types of cartilage. We're going to have hyaline cartilage, fibrocartilage, and elastic cartilage. So hyaline cartilage is going to be the most widespread type of cartilage. It is has abundant collagen fibers. Its locations are going to be places like the trachea. It's going to attach the ribs to the breastbone. It's going to cover the ends of long bones. And it's going to make up the epiphyseal plate or the growth plates of long bones. Elastic cartilage is going to provide elasticity. The location is primarily just the external ear. And then we also have fibrocartilage. Fibrocartilage is highly compressible. It is going to form cushion-like disc between vertebrae of the spinal column. We also have dense connective tissue. The main matrix element is going to be made of collagen fiber. Fibroblast, again, we talked about this earlier, but these are going to be the cells that make these fibers. We will find these in our tendons where skeletal muscle attaches to bones. We'll find them in our ligaments that are going to attach bone to bones at the joints, and are going to be these are going to be a little more elastic than tendons. And then dense connective tissue is also going to make up the dermis, which is the lower layer of the skin underneath that initial epidermal squamous layer. We can also have loose connective tissue. These are going to be soft. They're going to have more cells and fewer fibers than other connective tissues except for blood. This is going to be things like our areolar tissue, our adipose tissue, and our reticular tissue. So areolar connective tissue is going to be the most widely distributed connective tissue. It's going to be a soft, pliable tissue, kind of like cobwebs. It is going to function as a universal packing tissue and a glue to hold organs in place. Layers of areolar tissue called lamina underlies all of the membranes. And these fiber types kind of form a loose network. So it's going to, whenever you have swelling, this is the tissue that is soaking up extra fluid to cause that, because it is just kind of so loose and flowy. We can also have adipose connective tissue. And this is an areolar tissue in which there are more adipose cells than normal. So the function is to insulate the body, protect organs, and serve as a site to store ATP until we're ready to use it. This is primarily going to be in the subcutaneous tissue underneath the skin, and to help protect organs, especially like you'll find it around the kidneys, because those are really tender and really important organs. So around the kidneys, around the liver, that kind of thing. You also have reticular connective tissue. So what reticular connective tissue is a delicate network of like interwoven fibers. And this is going to have reticular cells like fibroblasts. So this is going to form the stroma, or the internal framework of organs, which can support blood cells or like lymphocytes. We will find this in the lymph nodes, the spleen, and the bone marrow. So let's talk a little bit about blood. We also call this vascular tissue. So blood cells surrounded by a fluid matrix, known as plasma, is how we make up all of blood. So the soluble fibers within blood are only going to be able to be seen during clotting. And the functions of blood is primarily to act as a transport vehicle for the cardiovascular system. It's going to carry nutrients, waste, and respiratory gases. Let's talk a little bit about muscle tissue. I know you all are probably getting really tired of this lecture. We're getting really close to the end. We just have about 10 slides left. So muscle tissue can contract or shorten to produce movement. Muscle tissue is what we call irritable, or able to respond to stimuli. We're going to have 3 different types: skeletal, cardiac, and smooth, and we looked at all of these in the lab this week. So skeletal muscle tissue is going to be packaged connective tissue sheets into skeletal muscles, which are attached to the skeleton, and they're going to pull on the bones and the skin kind of like levers. It's going to be voluntary, which means it's going to be consciously controlled. It's going to produce gross body movements or facial expressions. So the voluntary movements that we want to control that are very obvious when they happen is the layperson speak for all of this. Characteristics of skeletal muscle cells are going to be striations. So these are these little stripes that you see that make it kind of look like a fishbone type situation. They're going to be multinucleate. That means that each cell is going to have more than one nucleus. That makes it really easy to identify them under the microscope. And they're going to have a long cylindrical shape. Cardiac muscle tissue is involuntarily controlled. It's only going to be found in the heart, and its purpose is going to be to pump blood through the blood vessels. The characteristics, when you're looking under a microscope, of cardiac muscle tissue, it is also going to have striations, although they're going to be a little bit more spread out and look a little bit different. There's only going to be one nucleus per cell, so that's how you can differentiate it from skeletal tissue. And they're going to have short branching cells. So like you saw in the skeletal muscle how it just looked like long tubes, but they were all in a line, and you can see these kind of branch off in this way. You're also going to see intercalated disc, and what intercalated disc are these little gap junctions that are running between the cells. The purpose of intercalated disc is to coordinate the pumping of the heart so that it all pumps together when it's supposed to, and we don't have random muscles firing at different times, because that would make the whole system really ineffective. Smooth muscle tissue is going to be involuntarily controlled. It is found in the walls of hollow organs such as the stomach, the uterus, and the blood vessels. Peristalsis, which is that wave-like motion that you feel that is kind of like the feeling of digestion, is going to be a typical activity associated with smooth muscle. And characteristics of smooth muscle when you're looking at it under the microscope, it is going to have no visible striations, it is going to have one nucleus per cell, and it is going to be spindle shaped, which means each one is going to look very much like its own thing, as opposed to cardiac and skeletal tissue where it all looked a little bit smushed together. Let's talk a little bit about nervous tissue. Nervous tissue, its function is to receive and conduct electrochemical impulses to and from body parts. It is going to have it is going to be irritable, and it is going to be conducible. It is going to be composed of neurons and nerve support cells. The support cells are going to be called neuroglia, and they are going to insulate, protect, and help support those neurons. I gave you a little diagram here that kind of just goes over all of these slides that we just covered, all 20 or whatever it was of them in one little graphic there to make your studying a little bit easier. Let's talk briefly about tissue repair. So tissue repair is going to occur in one of two ways. You can either have regeneration or fibrosis. Regeneration is going to be the replacement of destroyed tissue by the same kind of cells. And fibrosis is going to be the repair by dense or fibrous connective tissue. Fibrosis is what happens when we end up with scars. So tissues that are going to regenerate pretty easily are going to be epithelial tissue, because again, that's always being sloughed off, and fibrous connective tissues and bones. Tissues that regenerate pretty poorly are going to be things like skeletal tissues. And tissues that are replaced largely with scar tissue when things happen are cardiac, muscle, and nervous tissue. So here are the events of tissue repair. First, you're going to have inflammation. So inflammation is really going to set the stage. The capillaries are going to become very permeable. Clotting proteins are going to migrate into the area from the bloodstream, and a clot is going to wall off the injured area. After that, you're going to enter the granulation stage. So at this point, you're going to get granulation tissue. You're going to get growth of new capillaries. Phagocytes are going to dispose of the blood clots and any leftover fibroblasts that need to be moved out. And then at the end of this process, you're going to start rebuilding those collagen fibers. Regeneration is going to be the last stage of this. And so regeneration and fibrosis are going to affect the permanent repair of this whole process. So at this point, you will either end up with a scab, and then that scab detaches and you're done with the whole process, or things are going to go a little sideways and you're going to end up with a scar and you'll go into that fibrosis type cascade. Let's talk a little. So the way that these cells are all going to grow and change and regenerate are going to be a little bit different depending on the cell type that we're talking about. So on this last slide, I just gave you a few examples that our textbook gives of different variations in how that process can go. And also at the bottom here, we talk about neoplasms, hyperplasia, and atrophy. So these are things that can go wrong in these cell replication processes and things. As always, if you have any questions or the content just isn't sticking, reach out. We will figure out a way to get the information to you and get you through it.